Right, so thanks everyone for tuning in, and thank you, of course, to the organizer for this really nice webinar initiative. So today I'm going to show you a bit of an unusual way of looking into large DNA systems by NMR using this Matthew-Chorley effect. So of course, in NMR, on one hand, we always like to brag about how biomolecular dynamics is accessible over a wide range of timescales. But on the other hand, up until recent years, we used to complain about how this dynamical information as well as structural information is often limited by the size of the macromolecule. But of course, with the introduction of high field magnets, as well as improvements in the way that we prepare our samples, and the way we conduct the experiments, particularly the introduction of Chorley techniques, and in parallel progress in the isotopic labeling schemes, now NMR is made amenable to proteins that exceed 100 kilodaltons in size. Now, all of this is true for proteins, but what about nucleic acids? So it turns out that we're a little bit behind when it comes to very large DNA and RNA systems. So if you take, for example, this classic protein DNA complex, the nucleosome core particle, which is the smallest repeating unit in chromosomes. So if you take the genome out of the cells and fully extend it, you will get this chain of beads where each bead contains a complex of proteins, and the beads are connected to each other through short and long stretches of DNA. Now, each of these beads is composed of 4 different types of histone proteins, H2A, H2B, H3, and H4. And each of these histones appear in 2 copies in the complex. Together, they form an octameric complex that is then wrapped by approximately 150 base pairs long DNA molecule, which is wrapped in nearly 2 turns of this double helix. So in this 200 kilodalton complex, we have nearly 10,000 proton atoms. So you can imagine that in order to get any kind of information, we really need to remove these protons and replace them with deuterons. But that's usually not enough if you want to get any kind of information, since most of the amides in this complex are not very visible by NMR unless they're localized to very flexible regions. So what we usually do is introduce methyl groups in the form of isoleucine, leucines, and valines into particular regions in the complex. In this case, I'm showing the labeling of the H2B histone. And combine this, of course, we keep the rest of the molecule deuterated. So then we combine this with a standard HMQC experiment where you get a high resolution spectrum that corresponds to this one protein in this big complex. So now we thought of, okay, so why don't we try and look into the DNA portion of this complex, since this was never done before by NMR. There wasn't any good way to look into this large DNA. And the way, luckily, we have a way that nature has given us to place methyl groups onto the DNA bases. So this involves this simple reaction, which has the methyl donor, the S-adenosyl methionine, and an enzyme called the DNA methyltransferase, which picks up this methyl group and places it, in this case, onto the C5 carbon of the cytosine base. So different methyltransferases recognize different DNA sequences, and they can place the methyl group onto 1 or 2 of the bases within this recognition sequence. And in fact, methyltransferases are known for their being part of the restriction modification systems, where for each methyltransferase, there's a corresponding endonuclease, which is an enzyme that recognizes the same sequence, but instead of methylating the DNA, it actually cuts the DNA. So this is one of the mechanisms by which bacterial cells actually protect themselves from bacteriophage infection, since the host DNA can be methylated and protected, while viral DNA, which is usually unmethylated, is quickly digested. So there are 3 ways. Two of them involve cytosine base. One can place the methyl group onto the C5 carbon, and the other one can place it onto the nitrogen 4 group, and this produces the so-called 5-methylcytosine and 4-methylcytosines. And the other modification involves the amine group of the adenine base. So of course there are other DNA modifications, but they're non-natural, non-native. And so these types of modifications are usually do not alter the integrity of the DNA. So all we need to do now is introduce the spin labels so we can look at this DNA by NMR. And so we developed a technique to do so that is a one-pot methylation reaction, which in the first step you have, you mix the methionine, which is labeled at the methyl group, and ATP, this in the presence of an enzyme called S-adenosyl methionine synthase, produces the methyl donor SAM, which carries the spin label, and SAM then reacts with the DNA in the presence of methyl transferase, which then, of course, places it onto the site of the base. So we monitored this reaction in the NMR tube, and for this we used a small oligo and 11 base pairs long that contained a recognition sequence corresponding to an enzyme called DCM. So as you can see in the HMQC spectrum, you get the two methyl groups from the oligo, and you can also detect the precursors, the methionine that decays with time as it is being consumed, and S-adenosyl methionine is being built up and consumed for the methylation reaction. So now that we saw that it's working, we realized that in order to be successful with this methodology, we really need to have the ability to produce as many methyl sites as possible. So for that, we tested a variety of methyl transferases. On the upper panel, you can see the cytosine modifications using 3 different enzymes. And the bottom panel involves the adenine methylation, again, 3 different enzymes. So there are 2 things I'd like to note here. One thing is that you don't need to perform the reaction separately. You can mix all the enzymes together in one tube and get the methyls that you want on the desired DNA. And another thing is that some of the methyltransferases can methylate 2 groups, one on the top and bottom strands of the DNA, and other methylases just introduce a methyl on only one of the strands. And also, because of the differences in the chemical shift between these 2 modifications, you can always combine cytosine and adenine methylation without compromising the quality of the spectra. So now that we had a variety of enzymes, we thought of, okay, so let's test it on the nucleosomal DNA, which is 153 base pairs long. So we picked up 3 methyltransferases. One of them is a cytosine methylase that you can see the spectrum here, the peaks here. And the other 3 perform adenine methylation, and we got a very high resolved spectrum even at room temperature and using moderate magnetic fields. So, so the next step was taking this DNA and wrapping it, the next step was asking whether or not we need a deuteration. And of course, we know with the proteins that it's essential to get a good spectrum, but with DNA, that was a question because you have a much lower proton density in the molecules. So in order to test that, we prepared 2 different samples, deuterated nucleosomal DNA and protonated nucleosomal DNA, and we compared the signal intensities of the 2 HMQC spectrum, and you can see that there is a significant enhancement in sensitivity for the deuterated case, and only a moderate enhancement for the cytosine is very significant, and for the adenine is only moderate. And so in order to understand where this comes from, we ran T2 relaxation experiments. In this type of experiments, we can select the slowly relaxing component of the magnetization. So the relaxation decay is mono-exponential. So from the fits, we can get the relaxation rates. And you can see that for the cytosine modified bases, there's a large difference in the magnetization, in the relaxation between the protonated and the deuterated samples. and whereas for adenine, the difference is smaller. And so this comes actually from, this can be explained if you look at the structures and the proton environment of each of these modifications. So we measured the effective distances between the protons of the methyl groups and the protons from the environment, and we saw that no matter which neighbor you have around this methyl group, the distances between, for the case of the cytosine methylation are much smaller than for the case of the adenine methylation. So as a general rule, we concluded that if you want to label their cytosine, you will probably need deuteration, while with 6-adenine methylation, you can get away with protonated DNA. So now we took this nucleosomal DNA and wrapped the nucleosome and prepared the nucleosome core particle. In this case, we also labeled the ILV residues of the H2B histone that is shown here in red. So this is the HMQC spectrum of the nucleosome core particle containing the protein portion as well as the DNA portion of the molecule. And what's an important outcome of this is that you do not need to choose whether you want to look at the DNA or the protein. You can look at both at the same time because the chemical shifts are well separated. So the next thing we did was trying to figure out what kind of dynamical information we can get from this type of systems. So the first experiment we ran was the proton-proton cross-relaxation experiments that from which we can obtain the order parameters, in this case for both the protein and the DNA methyls. So as you can see from this bar chart, for the protein part, you have a wide range of order parameters as expected because some of the protein regions localized to, some of the methyls in the protein localized to rigid areas or flexible areas. So you have a bunch of order parameters. And for the DNA, the distribution is rather uniform. And this can be explained by the fact that this DNA is tightly wrapped around the protein core, and which actually make these particles quite stable. So, so with this in mind, and we hope that the methodology can be further utilized to study and look into other potentially interesting aspects of nucleosomes. For example, how chromatin remodelers and regulators bind to nucleosomal DNA and perform basic cellular processes such as remodeling and gene regulation. And we can also now look into chains of nucleosomes and see the differences in dynamics between regions where there are linker DNA as opposed to regions where the DNA is wrapped by the histone particles, and maybe learn something additional about the systems, and this time from the DNA point of view. So, of course, aside from nucleosomes, we think and hope that this would be an opportunity, would give an opportunity to study other protein DNA complexes that usually involve very large DNA chains. Hopefully we'll see in the future. And with this, I'd like to acknowledge the people that were part of this work. Of course, Luis for his mentoring, and Al was a research associate in our lab. He was working closely with me on this project. And thank you for listening.
Thank you very much. We'll move straight on to the next talk by Enrico. So Julie, you may have to stop sharing and then Enrico can share. Julie, you need to stop sharing. Thank you.
So I start right away. Just a quick glance of what our center is. This is the Magnetic Resonance Center of the University of Florence. The numbers that you see are the proton-Larmor frequencies of the spectrometer, and you see the 1.2 there, which has been up and running since last year. What I will talk about today is a particular area of the research in metalloproteins. So you know or may know that several proteins can bind metal ions, and the metal ions can be either diamagnetic, like zinc or magnesium or calcium, or natively paramagnetic, like manganese, iron, nickel, cobalt, copper. In any case, even if you have a diamagnetic one, you can still substitute it for a paramagnetic one almost all the times. As always, when you have an unpaired electron, this unpaired electron provides an efficient relaxation mechanism for nuclei, and this wipes out the signals around the metal, creating the blind sphere. However, you have this is paid off by a large amount of structural and dynamic information encoded in the long range around the protein through pseudo-contact shifts and residual dipolar couplings, and this has been used since decades for obtaining the additional structural restraints in biomolecules. However, there is a growing need and interest to look inside the blind sphere to get to know the structural features of the active site, because the metal may be active for some for catalytic events. So how can we look inside the blind sphere? There are a lot of techniques like Mossbauer spectroscopy, but this is limited in the number of isotopes that can be looked at. There is EPR spectroscopy, which is a great tool for several metals like copper, cobalt, but has limitation of requiring often very low temperatures, and is particularly bad for integer spin systems where you need very high fields and very low temperatures in order to see something. Or you can rely upon Fermi contact shifts as observed in NMR, which are good for iron sulfur proteins, for instance, good for cobalt and nickel, but might be quite challenging for copper. The interpretation of data for all these techniques heavily relies upon quantum chemistry, and the models that have been produced for this to this end are growing continuously in quality. Still, you have faced the problem that the information that you can gather from EPR, from Mossbauer, or from Fermi contact shifts is limited to a small number of observables in the immediate vicinity or on the metal center. Whereas if you could somehow gather information from outside the blind sphere, also just on simple geometric grounds, you could have a much more robust and reliable information. So how can we look inside the blind sphere using long-range information? And it all comes from the pseudo-contact shifts, and the pseudo-contact shifts are a manifestation of the existence of the magnetic susceptibility of the metal center. And this magnetic susceptibility is like having a small magnet inside your biomolecule, which deforms the magnetic field as it's sensed by the nuclei. And this dependence is to a very good approximation, dipolar in nature. So this translates into the problem of calculating the contribution of the magnetic susceptibility. And if the magnetic susceptibility is at all the observable that is linked to the pseudo-contact shifts. Because when you do it from first principle, it seems that the magnetic susceptibility is not the real thing of interest. However, when you plug the experimental values into the equations that you get from different theories, you clearly get that the semi-empirical view is to be preferred over the quantum chemical treatment. So where's the catch? The catch is that the quantum chemical treatment was not complete. So we spent a lot of time trying to understand why. And the reason why turned out, and this is great collaboration with Lucas Lang and Frank Nese, comes from a gauge correction which was missing and restores the right symmetry in the problem. And so you basically we could basically conclude that chi is actually the thing to look at. We also did a little bit of verification of the methods to calculate the observables, checking also that the magnetic susceptibility is calculated in at least in our cases way better than the contact shifts contribution. And so we could finally move to the real problem. So we took a cobalt binding protein where cobalt substitutes zinc. The protein remains active with the metal substitution, but we inhibit it because otherwise it would self-cleave in a matter of minutes. And we first of all ascertain that what is the protonation state around the metal, because if the inhibitor, which is acidic, is assumed to be deprotonated, the agreement to the data is much worse than if you assume that the inhibitor is protonated. So we went on with the calculation with the protonated inhibitor. All the calculations were performed with ORCA at the CASA-CF and FPT2 level of theory, also including relativistic effects through the Douglas-Crowell-Hess Hamiltonian. And with this machinery at hand, this is the starting structure, and through 3 steps of steepest descent, we bring the disagreement between experimental and calculated to within the experimental uncertainty, with very minor changes in the overall geometry. If you really want to have to give a quantitative view on the changes that we created, is that the geometry is somehow getting closer to a trigonal B pyramid, which is quite good for cobalt indeed, but it's I wouldn't say that the cobalt is dictating the geometry around it. It's more likely the protein that dictates the overall geometry to the metal. And so, as a quick summary, as Frank Nese likes to say, to have fun with ORCA, we actually did have fun with ORCA, because it is possible to refine the structure of the active site of a metalloprotein using long-range PCS data, which is, to us, a very good result. Of course, the thing has some cons, because the procedure is slow. It's made slow by the relativistic calculations. Of course, we need to rely upon extremely limited structural models, so we basically neglect the existence of the protein itself, and then also the procedure has been implemented approximately manually. So there is an urgent need to make it at least automated in some way. So if you also want to have fun, you may like to come to us, because we have big magnets, nice projects, we have cookies, of course, and you can consider applying for IMBO, some Marie Curie or Instruct fellowships, and transnational access as well. With the acknowledgements, it's a quite long list, so I leave you to read it, and this is all from my side.
Thank you very much. So again, we'll keep the question till later. So our next speaker is Romeo.
Does everybody see my screen? Perfect. So morning, afternoon, or evening, everybody, depending on your time zone. I'd like to start by thanking the organizer for inviting me and giving me the chance to present my work here. Today, so my name is Romeo. I'm a PhD student in Petra Rovo's group at LMU Munich in Germany. And today I'll drive you through the dynamics of epigenetic DNA modifications by protein NMR with a specific focus on formylcytosine and carboxycytosine, especially within melting and base pair dynamics. So before we dive too much into the dynamics of those two molecules, I'd like to give you a bit of a spin on where do they come from. As Julie mentioned, they come from canonical cytosine, which can be methylated to yield 5-methylcytosine. Methylcytosine has then two choices essentially. It can either get deaminated to yield canonical thymine, or it can be stepwise oxidized by TET enzymes to yield 5-hydroxymethylcytosine, 5-formylcytosine, carboxycytosine. And those latter two, together with thymine, they are both substrates for TDG enzymes. And what they do is TDG enzymes cleave the nucleobase at the glycosidic bond right here, and yielding an abasic site, which then gets repaired to yield back cytosine. Apart from being protagonists in the cycle, both formylcytosine and carboxycytosine affect the radiant specificity of enzymes. They have peculiar patterns of distribution within certain cells in the genome. I'm talking specifically about stem cells and cancer cells. And they also have an impact on structure and stability of DNA double strands, most of which is either uncharacterized or still under debate. So those two papers here don't seem too much in agreement. So we think those systems were interesting, and the way we thought to study them, so the study rationale behind it, was to devise one model system, which in this case is a 12-methylpalindromic double-stranded DNA, where the sixth position can either be canonical cytosine or formylcytosine or carboxycytosine, because of three different chemical molecules. But because carboxycytosine does have a titratable substituent, we thought it would be wise to study that at least three different pH conditions. So let's say pH 7, 5.8, or 4.7. So to summarize, we have one single model system with three different chemical molecules, but 5 samples due to the fact that we have 3 of those pH conditions. And we studied that, I guess that by now, you know, given that you're attending this webinar, by NMR spectroscopy. So first things first, we started with assignment experiments, and here I am showing proton-carbon correlation spectra, HSQC specifically. And on the left, I am comparing canonical cytosine, formylcytosine, and carboxycytosine at pH 7. And you can appreciate how for most sites, the cytosines here or the guanines here, all those signals almost perfectly overlap. The ones that do not overlap are either the modified site, which shift by a very wide range, and also the nearby sites. So for example, this thymine 5. Very similar things can be said about carboxycytosine at different pHs, where the only sites that really shift are either the one directly on the modified base or nearby, but most of the rest of the ones are perfectly overlapping. Very similar conclusions can be drawn from proton-nitrogen correlation spectra. Here it's even a bit more obvious, perhaps. They all overlap exactly, irrespective of the condition, apart from the base pair, which involves the modified site. So G7 is the counter strand base pair of the DNA that we're studying. So we used all this nice chemical information to plot this in a chemical shear displacement study. And what can be drawn here is essentially the same conclusion, which is that only the modified site displays some chemical shear difference, which are very trendy too, so that's nice to see. But sites which are not directly at the modification are completely unaffected by either the modification or the pH condition, which means that the DNA is really, really tight, despite the environment being more acidic than normal, perhaps. So what we conclude for this is that structure is not really affected, at least in our conditions. So if structure is not affected, then we thought that dynamics might be. And NMR is the premier experimental technique for probing conformational dynamics in biomolecules. Today, we will specifically focus on block McCulloch exodispersion and CEST for probing micro and millisecond timescale. Another thing that needs to be acknowledged is that DNA is a prime target for dynamic studies, given that it entails essentially two big timescale motions. The first one, the fast ones between nanosecond to femtosecond, which involves breathing, bending, twisting, etc. And the slow ones, which is base pair opening, melting, and annealing, etc. One timescale which appears to be missing entirely, at least in healthy, non-abasic, non-damaged DNA, is the microsecond timescale one. So it hasn't been reported, at least in normal conditions. Given that we are working with strictly unlabeled samples, we use the Salopi methodology from Petzl Lab, reported in 2018, just a few years ago. Very, very handy to work. Both pulse sequences for ARMRO and CEST are displayed here. They are extremely similar. They both start with creation of selective coherence. Then there is a spin-lock element. We have the evolution of the indirect dimension. Then we have coherence transfer elements, and then we have water suppression by excitation sculpting, followed by acquisition. As I mentioned, the only difference involves the two spin-lock elements. In the ARMRO, we probe transverse plane magnetization. In the other hand, in CEST, we probe Z-axis magnetization, which is the reason why CEST can access slower timescale motions. Z-axis relaxes slower in general than R2 transverse plane magnetization. On the right, you see typical Salopi spectrum. This is irrespective of whether this is ARMRO or CEST or without the spin lock entirely. On the diagonal line lie all the peaks which do not have a 3J coupled partner, so that this pulse here is essentially useless for them. But instead, the cytosine that have H5 and H6 3J away from each other, then they get displaced as cross peaks. And here I'm showing some selected textbook two-set exchange, which we measured for this DNA system. This is comparing orange from the cytosine and canonical cytosine in blue. And what we're probing here is the equilibrium between double-stranded and single-stranded DNA. So it's a melting profile. The reason why we are so confident in assigning this to a melting profile is that the excited state here, so there will be the alternative conformer, the single-stranded DNA, chemical shift is almost exactly what we expect from the neighbor corrector Raman-Cohen chemical shift values, which is very nice to see, calculated from D-Shift. It's a very handy software from Lab and coworkers. And the second hint is that by making the temperature higher, the size of the secondary dip grows. So that's a pretty definitive indication of that. What does CEST provide us apart from chemical shifts? It does provide exchange rates and populations of those two states. So how can we use this exchange rate and population to get more information only from CEST, so the same experiment? Well, having the exchange rates, having the population in the ground state, that will be the double-stranded DNA, having the concentration of the total monomer in solution, we can discern association and dissociation kinetics, and we can then cross-correlate them, yielding some interesting observations. So here we are still comparing 5' cytosine with canonical cytosine. We are cross-correlating the dissociation kinetics of the 5' cytosine modified sample with the canonical one. And in those plots, as slope of 1 means that the rates are unchanged, meaning that 5' cytosine wouldn't have any impact on DNA melting or annealing. Higher than 1 means faster kinetics, and smaller than 1 means lower kinetics. So we can see that because the slope here is much bigger than 1, far from the cytosine facilitates DNA melting, and at the same time, it makes annealing ways lower. So it's a combined effect. We can further elaborate on this K and KDs by using some physical chemistry. And we can use the relationship of dissociation and association kinetics together with the temperature in order to plot them into Van 't Hoff and Arjen Polanyi formalisms. So here in the middle, we have those profiles, which are derived from those two formalisms from the same sites that I showed you before, T5, C6, and A8. In the very middle, we see a potential energy surface derived by thermodynamic and kinetic activation parameters from those two frameworks. And we can see that this stabilization, which is induced by far from the cytosine, actually only matters for the double-stranded form. There is no energy difference when it comes to transition state. And we actually see that every single site that we analyzed, I'm not showing them all here, but you can check the supplementary information on this paper here to see that all the association kinetics work in an anti-Arrhenius behavior manner, which means that they are inversely correlated with temperature, and the opposite can be said about the dissociation kinetics. The only site which did not obey this behavior is the formal proton on the far from the cytosine modification, and we thought this was interesting. Unfortunately, we have no comparison for canonical cytosine because there is no formal group on canonical cytosine. And both of them displayed an Arrhenius behavior. And we thought this was because we have a very strong intermolecular hydrogen bond here, which means that in order to break this, and make this rotate, we need to apply more and more energy. So we thought that this proton does not actually report on melting, but it does instead report on the amount of energy that we need to break this intramolecular hydrogen bond. So what is the mechanistic relevance for far from the cytosine melting at all? So comparing canonical and from the cytosine, and carboxyl is in the works, we still have preliminary data only. We know that the way DNA melts is that it unzips starting from the fraying ends. So from here, and what we think from the cytosine does is introducing a weak link in the middle, and this helps melting. Essentially, it's a weak link on a chain. You can imagine that way. What we especially speculated, but we didn't know, we had no experimental results before, is whether this weakening here actually introduces any fast timescale motion on the sites. So if there is any internal fraying on the microsecond timescale at all. And this is something that actually we checked very recently. We ran some overall oxygen dispersion experiments. And on the left, we are comparing far from the cytosine and cytosine, canonical one. And on the right, we're comparing carboxyl cytosine at different pH conditions. And what we can see here is a black line indicates non-exchange model, meaning that this profile does not indicate there is any conformational exchange going on, while a colored line means that the data fits better to an exchange model, meaning that there is microsecond timescale motion. Far from the cytosine does indeed show that, and it also shows a very interesting low R2 rate, up to 1 second, which is very, very small. Instead, carboxyl cytosine at pH 7 and 5.8 seem to behave exactly like cytosine, so without showing any conformational dynamics. Instead, whenever carboxyl cytosine is sufficiently protonated, it shows a very large exchange rate contribution to the R2 and displays microsecond timescale motion. So with this, I would like to thank everybody for your attention, and also my PI Petra and my students Thea and Pia, who were really instrumental for this study. And also I would like to thank our collaborators from the Corel group for providing us with the samples and interesting biological discussion. Thank you, everybody.